Welcome to the deep dive.
Eh.
So you've brought us a pretty dense stack of sources today. It's all centered on this huge new oncology study,
Mm-hmm.
a deep dive into the whole genome landscapes of, what, over 1,300 breast cancers from the Shubrick's cohort in Korea.
That's right. And this is, it's truly foundational research. We're talking one of the largest, most comprehensive efforts using whole genome sequencing, WGS, and deep transcriptome data, all on one clinically annotated cohort.
It's not just a map anymore.
No, not at all. It takes breast cancer research from a simple map to, well, a three-dimensional model.
Okay, so our mission for you today is to cut through all of that genomic complexity. You know, the copy number alterations, the structural variants.
All the jargon.
Exactly. We're going to give you the key takeaways.
Yeah.
We're hunting for the biggest insights that are, actively reshaping how we think about diagnosis, treatment, and maybe even prevention.
And we have to start with the context of this cohort. It's really important because it frames all the results. This group had a significantly younger median age, around 44, which is much younger than most Western studies, and a lower prevalence of the generally less aggressive ER-positive or luminal A subtypes.
And that demographic difference matters. Why? Exactly.
It's critical. It means the findings here give us new insights into more aggressive forms of the disease. It's essential for a truly global understanding.
Okay, let's unpack this then. I think the most mind-bending thing from these sources isn't about what mutation they found, but when all this molecular chaos actually starts.
Yes.
We tend to assume cancer develops over a few years, but this study, it suggests we're talking about decades.
That is the stunning conclusion of their molecular timing analysis. The researchers used the WGS data to trace the timeline of genomic instability, and they were specifically looking at copy number alterations, or CNAs.
So these are the big chunks of DNA that get, what, deleted or duplicated?
Exactly. And what their molecular timeline revealed, well, they found that the crucial period for acquiring these frequent, long segmental CNAs,
The really major structural damage.
right, the stuff that signals deep instability, it occurs incredibly early. These huge changes are already in place by just 20% of the total molecular time that the cancer has evolved.
And that's before the main tumor cell ancestor even shows up.
It precedes it, critically, yes.
So let's translate that 20% mark from, abstract molecular time into a human life. What are we talking about here?
We're talking decades. It implies that the genomic instability setting the stage for the tumor, that fundamental vulnerability, emerges decades before diagnosis.
Wow.
The authors even suggest this first big wave of genetic chaos could be acquired as early puberty.
That's unbelievable. So we have to stop thinking of this as a fast, sudden event.
Unequivocally, it is a multi-decade evolutionary process.
Wow. That just fundamentally challenges every timeline we have about cancer.
Oh.
I mean, if the damage that fuels the whole process starts when someone is 12 or 15, what does that even mean for prevention?
It shifts the entire research focus backward.
You're looking for footprints that precede any cellular changes by 30 or 40 years.
And we have specific evidence of this. They track the timing of the most common CNA, which is an amplification at 8q21.13. That region notably includes the MYC oncogene.
Ah, MYC, the classic cancer accelerator. So when was that one acquired?
At a relative timing of just 0.15, so extremely early in the tumor's history.
And does that early event actually matter in the clinic decades later?
It absolutely does. The clinical implication backs it up. Having that early MYC amplification was associated with significantly poorer overall survival. We're talking a hazard ratio of 1.67.
So it's not some irrelevant early step.
No, it's an early step that dictates the tumor's aggression 30 years down the line.
It's just amazing how early the chaos starts. But okay,
Yeah.
Once that chaos is underway, what are the specific weapons the cancer uses? Let's shift from timing to the mutations themselves.
Sure.
We all know the classics, TP53, PIK3CA. How did this deep WGS expand that roster of known drivers?
This is where that extensive sequencing was just so essential. It helps you find the truly rare but recurrently active players. The study identified 41 breast cancer driver genes in this cohort.
And 4 of those were completely new.
4 were entirely novel driver genes. Yes, they hadn't really been firmly established as breast cancer drivers before.
So who are these new enemies?
They are BCL11B, RAB1, RAF1, and SPECS1. And, the utility here is that we can now look beyond the usual suspects. For instance, BCL11B is involved in chromatin remodeling.
How DNA is packaged.
Right. And RREB1 regulates critical signaling pathways like RAS and TGFA. Finding these provides new targets, new intervention points.
So we have the timeline, we have the individual mutations, but the sources also talk a lot about the messiness of the tumor, not just what's broken, but how different all these cells are from each other. Intratumoral heterogeneity.
Correct. And with WGS, you can actually quantify that messiness using something called the MAF score, mutant allele tumor heterogeneity.
And this is where TP53 comes back in.
This is where it comes back. Tumors with TP53 mutations almost universally had higher genomic instability. They had more chromosomes, higher ploidy, and increased homologous recombination deficiency, or HRD.
So naturally, they would have higher MAF scores, more chaotic.
Much higher.
And can you actually use that score? I mean, does a high MAF score predict a worse outcome for the patient?
It acts as a powerful prognostic factor, especially in this context. They found higher math scores were strongly associated with poorer overall survival, particularly in that subset of TP53 mutant tumors.
How strong was the link?
The hazard ratio was 1.66, so a significant increase in risk. It tells you that the cancer's ability to evolve quickly, its diversity, is a measurable clinical threat.
If you're genetically unstable and constantly throwing new options at the wall, you're much more likely to find a way to survive.
That's the evolutionary logic, yes.
Okay, let's move to structural chaos, because this is just a fantastic example of cancer's engineering prowess. We're not talking about little point mutations now. We're talking about the genome physically tearing itself apart and stitching itself back together.
The structural variants, the SVs, are where the whole architecture changes. They cataloged over 208,000 of these events.
And most were local.
The vast majority, 78.2%, were local intrachromosomal rearrangements, basically reshuffling genes on the same chromosome.
But the really clever insight, the true engineering hack, that comes from the distant connections, right? When the cancer uses these SVs to create brand new regulatory pathways.
Exactly. And this study provided one of the clearest demonstrations of what we call enhancer hijacking.
Enhancer hijacking.
They found a recurrent interchromosomal translocation. It involved the ERBB2 locus on chromosome 17. The SV effectively moved a powerful piece of regulatory DNA, a super enhancer from chromosome 20, and dropped it right next to the ERBB2 oncogene.
So if the gene is a light switch, the enhancer is like a volume knob.
Of a very powerful volume knob.
And the cancer moved the most powerful volume knob it could find right next to its favorite oncogene.
That is a perfect analogy. And the result was significantly higher ERBB2 gene expression. And this is without necessarily duplicating the gene itself massively. It's a highly efficient shortcut to activate the oncogene.
We also have to touch on gene fusions, when two different genes get spliced together. I thought these were pretty rare in breast cancer.
They are generally rare, but the study highlighted some recurrent ones that are clearly important. In luminal cancers, they found imipel-1 TTC6 and the well-known CCDC170 ESR1 fusion.
And that last one is linked to therapy resistance, right?
Endocrine therapy resistance, yes. And for the basal-like cancers, they found a fusion, BCL2L14 ETV6, that's associated with increased invasiveness and resistance to the chemopaclitaxel.
That structural complexity is just stunning. But speaking of amplification, when a piece of DNA gets copied hundreds of times, we usually picture it just, stuck on the chromosome. But you found that a completely different mechanism is driving the most aggressive amplifications.
Yes, we're moving into the realm of extrachromosomal DNA, or xDNA.
Okay.
For over 40% of the most prominent focal amplifications, we're talking ERBB2, CCND1, FGFR1, the primary mechanism was the presence of xDNA. Overall, nearly 3 in 10 cancers in this cohort carried it.
So why is xDNA better for the cancer's evolution compared to just duplicating a gene on the chromosome?
Because xDNA are these small circular pieces of DNA that operate independently of the main chromosome. They can be massively duplicated and then distributed unevenly when a cell divides.
Which gives it an advantage.
A huge advantage. Two, really. First, massive rapid amplification. And second, they can be randomly jettisoned or acquired. It means the cancer can evolve its gene dosage much, much faster under pressure from therapy. It's the ultimate tool for rapid, flexible evolution.
That's critical. Okay, let's tie all this deep genomic info directly to the clinic. Let's take HER2 positive breast cancer.
Sure.
We usually diagnose it with protein detection, like immunohistochemistry, IHC. How does WGS data refine our ability to predict if a patient will actually respond to aggressive anti-HER2 therapy?
WGS boosts the precision significantly. Traditional IHC is sensitive, sure. It picks up nearly all the responders, about 95% sensitivity.
Right.
But WGS lets us measure the exact degree of amplification, the absolute copy number. And the study found that using a WGS-derived ERBB2 copy number of 33 or more, that showed much higher precision and specificity in predicting a complete response.
Wait, hold on. So you're saying a tumor could look HER2 positive by the standard test, the IHC3 plus A gauge, but it might not respond if the copy number by WGS is lower than 33.
That's what the data suggests. It's a move from a simple yes-no diagnosis to a quantitative prediction. It seems the tumors with the truly massive gene overdose, the 33-plus copy number ones, are the ones most addicted to that HER2 pathway.
And so they're most likely to be completely wiped out by the therapy.
Exactly. And it gets better. The predictive power was dramatically improved when they added another structural chaos marker, chromothripsis.
The catastrophic event where a chromosome just explodes and gets put back together wrong.
Precisely. When they factored in both the high WGS copy number and the presence of chromothripsis, the prediction was maximized. 71.1% of patients who got a complete response had evidence of it.
So a tumor under extreme structural stress is actually more vulnerable to that kind of aggressive therapy.
It seems so. It's a phenomenal biomarker combination.
But I think the most nuanced and maybe confusing clinical finding here is about homologous recombination deficiency, or HRD. We often see it as just a negative biomarker, but this study shows its effect is totally dependent on the drug you use.
This is the critical paradox you need to take away from this deep dive. We've always thought of HRD, the inability to repair DNA correctly, as just bad. But this shows it's a Janus-based predictor. It depends entirely on the therapy.
Okay, let's break down the two scenarios. First, chemotherapy in triple-negative breast cancer, TNBC.
Right. In TNBC patients getting adjuvant chemo, HRD was actually a positive indicator. Those deficient tumors are highly sensitive to DNA-damaging chemo. They showed significantly superior disease-free survival.
How superior?
An exceptional hazard ratio of 0.10. The genomic defect literally makes them vulnerable to the poison.
Okay. But then you look at hormone receptor-positive tumors treated with targeted drugs like CDK46 inhibitors, and the effect just completely flips.
It's the inverse scenario. In those patients, high tumor mutation burden and high HRD scores were associated with worse progression-free survival. HRD status was the single most significant negative predictor.
With an alarming hazard ratio.
10.20.
10.2, that's huge. That means HRD status is highly predictive of failure for that therapy.
Absolutely. 85% of the HRD patients in that group progressed on the CDK46 inhibitor. And they also noted that PTN mutations were exclusively present in the patients who progressed.
It just tells you that precision oncology has to move beyond simple mutation lists.
You have to consider the genomic context, the drug mechanism, and the tumor subtype all at once.
A powerful lesson. Let's end by looking at resistance and moving the genome from a static map to a history of treatment escape. WGS timing can show us this evolution in action.
It can. They noted that the acquisition of those large segmental CNAs actually accelerated after a cell acquired the HRD phenotype. Once the DNA repair machinery broke, the cell just ramped up its structural chaos.
Instability feeding more instability.
Exactly. And there's this phenomenal, almost unbelievable case study of a tumor finding an escape hatch. Patient 703 and the rescuing mutation.
Tell us about this.
So this was a patient with a germline 1 base pair deletion in BRCA1. Because of that, the tumor was highly HRD and was being treated with talazoparib.
A drug designed to exploit that specific weakness.
Right. The drug damages the DNA, and the BRCA1 deficient tumor can't repair it. But the tumor fought back. Post-therapy, WGS revealed an 8 base pair somatic deletion that, just by chance, overlapped with the existing germline deletion.
So a second mutation came in and acted as a functional repair. It fixed the problem the drug was targeting.
Precisely. It was like the cancer found a way to bridge the two breaks caused by the first mutation, soldering the reading frame back together. This combination restored BRCA1 function. It converted the tumor back to being homologous recombination proficient, or HRP.
And the targeted therapy became useless.
Useless. And the timing analysis was vital here. It showed this rescue mutation was acquired relatively late, around 50% of the molecular time, right when drug pressure would have been highest. It's a perfect illustration of evolution in action.
So what does this all mean for you, the listener? WGS gives you a level of detail that is just indispensable. It goes so far beyond targeted sequencing. It's finding novel driver genes, revealing these unexpected mechanisms like enhancer hijacking and AKNA. And most importantly, it's forcing us to refine and rethink clinical biomarkers like ERBB2 copy number and the paradoxical function of HRD status.
This robust genomic platform allows for the necessary integration of clinical outcomes with this really granular genomic data. We're moving away from simple static snapshots. We can now track a cancer's history, understand its future moves toward resistance, and apply therapies based on a dynamic, three-dimensional view of the disease.
It's the foundation of truly precise oncology.
It is.
It paints a picture where this disease is a decades-long evolutionary arms race.
Yeah.
So before we wrap up, here's a final provocative thought for you to chew on, connecting this specific cohort back to global health.
Okay.
This study found that a germline deletion of ACOBIC3AB, which is associated with massive hypermutation in tumors, is significantly more common in this Korean cohort. It's present in almost 32% of them.
Compared to about 8.5% in European population.
Right. So since this germline difference is tied to a higher overall tumor mutation burden, how might these distinct population-specific germline backgrounds influence global strategies for cancer screening and prevention? Maybe decades before we could even see a tumor.
That's a great question.
I'll leave that with you.